Good afternoon, everyone. Welcome to today's webinar on Droplet Digital PCR technology, hosted by Lasec in collaboration with Bio-Rad. In today's webinar, we'll dig into the fundamentals and application of ddPCR, a groundbreaking tool that offers precise and absolute quantification of nucleic acids. Whether you're a researcher, a professional seeking to expand your knowledge, or someone with a keen interest in genomic technologies, this session is designed to provide you with valuable insights and practical knowledge. We are pleased to have Luke Forbes with us today as our speaker. Luke is a distinguished field application specialist from Bio-Rad with a strong background in genomics and digital PCR. His expertise spans the quantification of nucleic acids, detections of rare mutations, and gene expression analysis. Luke is dedicated to advancing genomic technologies and frequently collaborates with leading experts to bring the latest innovations and best practices to our region. Before I hand over to Luke, kindly note that the chat function is disabled. However, I would like to encourage all participants to use the Q&A feature to ask any questions you might have. So without further ado, please join me in welcoming Luke Forbes. Over to you, Luke.
Great. Thanks so much, Michelle. Thank you for the introductions. Lovely to be on this meeting in the middle of a Thursday. Thanks for making time. So to get started, I think we can get straight into it. There's quite a lot to discuss. I've had to keep it quite short. But today, basically, I'm just going to give you sort of an overview of the technology, how it works, where it falls, and sort of what you can do with it. And then if there's any questions, we can go into much more detail, or you can take it offline, we can discuss it further. Or you can join a webinar that we'll be hosting in the future, which I'm sure there will be still quite a few. Great. And remember, sometimes that there's really no silly questions. Please feel free to ask any questions. I've probably heard most of them by now. So there's no bad questions to ask. Please feel free to ask anything. It always helps, and it helps engagement as well. So thank you very much. So when we describe ddPCR, so ddPCR stands for Droplet Digital PCR. When we describe it, we're really talking about a third generation in PCR. So it's built on the foundations of PCR in general. The first type of PCR in 1985 was qualitative PCR. That's the one where you run your PCR on a thermocycler, in an endpoint thermocycler, and you get a yes/no answer. You just get bands when you run a gel, and that just gives you a presence-absence detection test. Still really commonly used and a great technique, but has some limitations. As you know, there's no quantification method, and sensitivity can be a bit lacking as well. Then we've got the second type of PCR, which became very popular in recent years, especially with COVID as well, which is real-time PCR. The difference between real-time PCR and endpoint PCR is obviously the fluorescence addition. So the fluorescence is the method of detecting your amplicons of interest. But it didn't just change the detection with fluorescence, it changed the detection point. So in endpoint PCR, you're detecting your results at the end, but in real time, you're detecting it at the exponential phase. And that's why in real-time PCR, you need to do a lot of things to make sure that your results are accurate. Some of those things are looking at your E-value or your efficiency. That's just a measure of the doubling each round, just to make sure that your reaction is doubling as it should in a PCR reaction. You're going from 1 copy to 2 to 4 to 6 to 4 to 8 to 16, etc. And without that efficiency measure, you actually can't accurately quantify your amplicon or your target of interest. And another distinctive feature in real-time PCR is that you have to use a standard curve. Why do you have to use a standard curve? Because real-time PCR is just only relative. It's only relative. An unknown has only got a relative quantification or a relative amount compared to a standard or a known concentration or known amount. And now what you're here for today is to discuss the third technology, which is this Droplet Digital PCR, which is an absolute quantification method. And like endpoint PCR, the detection point is at the end of the cycle. So it's read at the end of the cycle, unlike real-time PCR, where it's read in the exponential phase. But like real-time PCR, it makes use of fluorescence tags or molecules that are bound. So some of you may be interested to know that the term digital PCR was almost described at the exact same time as the release of real-time PCR. It was almost slightly before in some aspects, because the methodology made a lot more sense than real-time PCR, because we were already using endpoint PCR. So it made sense to keep the detection method at the end, because no one wants to read the detection method in the middle. But there was huge limitations back then in terms of, one, the partitioning or the dividing of your sample. There was no accurate method to keep dividing your sample. And two, like many things back in the 1990s, there wasn't enough computer processing power really to make this technique very viable as well. And Bio-Rad actually was one of the first companies to sort of commercialize this technology. And Bio-Rad's had this technology on the market now for around 10, 11 years. So we're quite comfortable with the technology, where its benefits lie, where its limitations lie, and how it can really help improve your work. And we're going to discuss some of those benefits a bit later on. Now, when we talk about Droplet Digital PCR, ddPCR, when we talk about any digital PCR really is made up of two important steps. And the first is the digital step, which we'll discuss in a little bit. And the second is the partitioning step, okay? What makes Droplet Digital PCR slightly different to traditional digital PCR that you'll see from other manufacturers? Well, it's that we're using a droplet, a water and oil emulsion, which is creating a droplet. Now, these droplets, basically you have a standard qPCR reaction or a PCR reaction, and you're dividing a single reaction of around 20 microliters, and you're dividing that reaction into around 20,000 divisions, okay? So you're almost doing 20,000 little PCR reactions inside of one PCR well. Now, what is the benefits of using droplets as opposed to using a microfluidic plate or something like that? Well, the first is that the droplet volume can be validated. We validated this both internally and externally, and customers can also validate themselves. They can always even take samples and look at the partitions under a microscope, and you'll see they're all the exact same size. We're also validated externally. There's the national reference labs in Australia that have checked the volume of our partitions. Now, why is that such a big deal? Why is accurate partitioning very important? Well, because really when you're doing digital PCR, you're only just checking the number or the quantity or the count of the partitions. And if your partitions vary in size, your accuracy is never going to be correct. So that accuracy of the partitions is really important, that the partitions have to be the same size. Now, in Droplet Digital PCR, we're the only digital PCR system that has validated sizing on our droplets. We're the only one that can monitor them in real time as they go through the system, and we can reject any wrong or sized droplets, which is a huge benefit or key aspect of this. And we actually used to think the droplets were 0.850 nanoliters each, and we eventually came to, after quite a lot of research, to the accurate sizing of 0.795 nanoliters, which is really, as you can imagine, really granular detail about the exact sizing. So I always like to explain the technology to customers in terms of a workflow, because, you know, really as scientists, many of us can only really visualize or understand something. We're not in physics. Most of us on the biological sciences field, and we really like to understand something by seeing it or trying to understand how it sort of plays out. So everyone should be fairly familiar with the first step, which is that preparing a reaction. What goes into a real-time PCR reaction? Well, you've got your nucleic acid, or your sample of interest. You've got your primers that are specific to your amplicon that you're looking at targeting. And you've got probes potentially if you're using probe-based chemistry, or if you're not using a probe-based chemistry, you can use an EVA-based dye, which is like Cyber Green, similar. You've also got your supermix, which is specific to digital PCR. Why is it specific to digital PCR? Because the viscosity is just slightly different, because obviously you're dividing everything into a lot of droplets. So if it doesn't occur that it's in the droplet, no reaction will occur. And that's basically it. Your supermix is obviously composed of DNTPs and buffers and coenzymes, all that stuff that's normally contained in a supermix. And that's it. There's nothing else that goes in there. So when you're doing droplet digital PCR, really, it's basically like doing real-time PCR. In fact, you can use real-time PCR assays most of the time. And with the Bio-Rad system, we're running it on a 96-well plate. It's a standard 96-well plate, something you should also be very familiar with if you do any sort of PCR. Now, this is the difference. That 96-well plate is then required to generate droplets, okay? And there's two methods of doing this. You've either got your manual droplet generation, where you take 8 wells at a time, you move them into a cartridge, and you mix them with oil, and the system partitions for you, which takes around 2 minutes for 8 samples. So you get around 20,000 droplets or partitions per well for all 8 wells in 2 minutes. Or you can do it with an automated system, and the automated system obviously just takes away the hands-on time with that sort of procedure. You just select the number of wells you want to be partitioned, and it just does the pipetting automatically. Then once you've got your droplets generated, you take your generated droplets, you move it into a thermal cycler. This is a standard thermal cycler, probably one you already have in your lab, or you can use one that you're purchasing, and you do the thermal cycling process. That's a standard thermal cycling process. And in that process, some of the partitions that contain your gene of interest or your target of interest, the PCR reaction occurs, primers bind, goes through the cycles, and fluorescence increases and fluorescence occurs. And for the droplets or the partitions that doesn't occur, they stay unfluoresced because there's no gene of interest. Then you remove your plate from the thermal cycler, you move it into a droplet reader, and inside the droplet reading instrument, the droplets are picked up in a single file. And this again is a very big key difference between droplet digital PCR and other digital PCR technologies. In droplet digital PCR, it's like a flow cytometer. So in a flow cytometer, you pick up your sample of interest, and your cells are each read by lasers one at a time, and they fluoresce or excited, and then they return with another wavelength, but they're read in a single file one at a time. Now that is the same for ddPCR. So each well, all 20,000 droplets plus minus, are read one at a time through a detector. In other digital PCR technologies, many of them will use a single camera and take a picture of all 20,000 on a full plate, or that can only go to 16 normally. So on a 16-well plate or a 24-well plate, and that's all looked at once. Obviously, when you do it that way, you're running into a bit of danger of things like crosstalk between channels and other issues that may sort of occur. So really, the big 2 points in any sort of digital PCR workflow is, 1, what is the partitioning method? And 2, how are they reading their samples? Are they reading it in one giant picture, or are they reading it individually as a single partition each? And in the case of the Bio-Rad system, we're reading it one at a time, like a flow cytometer. Now, remember I said to you, digital PCR has two distinct features over other PCR methods. 1, it's similar to real time, it has fluorescence. 2, it's similar to endpoint. It's at the reading is at the end of the cycling process. And then I said the differences is 1, in the partitioning. In real time and endpoint PCR, you don't partition your sample. but in digital PCR you do. So the partitioning method is very important. And the second is that digital signal. What does it mean? What does a digital signal actually mean? Well, anyone that knows anything about computers, digital just implies ones and zeros. It means that something's either 1, a positive, or 0, negative. Computers don't know the color green. They just see a series of ones and zeros after each other, and they know to recreate the color green. If you type in something to a computer, it converts it back to ones and zeros, and then it spits out an answer that you've given as a predefined answer for that sort of code. Now, the same thing is happening in digital PCR. Your values or your droplets are being assigned either as 1, positive, or 0, negative. And if you look at the example that's given here, remember these little droplets are running through a detector one at a time, and they're each being excited one at a time. And if they contain your gene of interest, i.e. your primer is bound in your probe, then they fluoresce in that color, and depending on the channels. You can have 1 or 2 channels or 5 channels or more, but each of these are inspected one by one, and then for each fluorescent channel, they are given a value as either positive in this direction or negative, no fluorescence or some fluorescence. And then they are their threshold is put between them, and it's very important that there's a good separation between your positives and negatives, and this threshold just divides them. And then what happens is it gets stored as a total number of negative droplets versus a total number of positive droplets, and that gets put into what we call Poisson distribution. Every single digital PCR system that is on the market today makes use of Poisson distribution. Why does every system make use of this sort of modeling technique? Well, because this modeling technique sort of it helps us to know how close we are to the absolute quantification. Remember, if you're only assigning a value of 1 or 0, what happens if a partition contains 2 of your gene of interest or 3 of your gene of interest? It's just random chance. It's just a division. So if that occurs, you need some sort of statistical modeling method to give an idea of what is the likelihood of you having more than 2 copies in a single droplet. And as your concentration increases, so does the likelihood of the number of genes increase. So the way I describe this to customers, generally, I'm sitting in an event sort of seminar type setup, and there's various tables. Maybe there's 10 tables. And each table there's 2 people, or there's 3. 2, 3, 4 is a mix of people. But if I count those 10 tables and I average them out, I can get an idea of how many people are sitting at each table. And if I had to add another 10 tables, I wouldn't be so accurate, but I probably wouldn't be far off. Now, if I had a sample size of around 20,000 tables, then I could kind of more accurately predict the likelihood of having 1 or 2 or 3 people at each of the tables going forward. It's just a method of fitting the data. Now, the good news is you don't have to do anything with statistics. It's not like real-time PCR. There's no interpretation. There's no manual delta CQ inputs and means and all those kind of things. It's just done in the background, where it just takes the fraction of positives and the fraction of negatives, and it's calculated. And in the software, it gives you the results. It says you've got 18 positives, you got 18,645 negatives. Those are your total number of accepted, and that's your concentration copies per well. And you can also see here in this example, this is well D06, and you can see these are your positives, and these are your negatives, and this is your threshold in between. Okay, so quite straightforward, really easy to work with. And you can't really introduce any bias, unlike in real-time PCR and things like that, because you can't say if it's over here, it's a negative, because it's clearly not. It's separate from the positives and negatives. Okay. Great. So I understand ddPCR. I understand Droplet Digital PCR to a degree, but how does it help my research? Well, do you like running standard curves? I assume probably not. Do you enjoy having low reproducibility? What do I mean by that? I mean, if someone runs a real-time experiment in this lab and then they run it in a different lab with a different supermix or a different system, they might get some slight different answers. It generally occurs. Do they have failed experiments due to inhibition, especially if you're working with something like wastewater or FFPE? Do you enjoy doing that statistical data analysis when you're ready to publish and they tell you, no, your efficiency isn't right, your R-squared values isn't correct, you need to redo this? What is your ANOVA information? What about looking at a technique like NGS where you have to, you guys know it's obviously quite expensive. There's also generally many specialists involved in your analysis sort of portion. And, you know, just to expand on this briefly, NGS is a really great tool for genomics, of course. I absolutely think it's a fundamental requirement for genomics. However, NGS is only going to give you a lot of information on something, but once you have a large quantity of information about something, you actually need much more granular, smaller details about changes within that thing. And that's where Droplet Digital PCR comes in. Really, it's not a competing technology, not at all. They're two totally different technologies. In NGS, you're trying to find something novel or find a lot of information about a whole sequence. In Droplet Digital PCR, you're trying to quantify that information, see how changes affect its activity in the cell or something along those lines. Okay. But in Droplet Digital PCR, because it's just a type of PCR, the cost is much less than NGS. Also, the data analysis is much easier. In fact, the data analysis is a lot easier than even just real-time PCR. So I made a lot of bold claims. What can I say about those claims? Well, the first thing is in ddPCR, it is yes, it is more precise than qPCR. There's a 10% discrimination. There's a much lower CV values, and your confidence interval is high, just because of that partitioning, which we'll get into. There's also another huge benefit, absolute quantification. It's sort of stated in the name. But in that, you don't ever have to run a standard curve, and you don't have to have standards unless you're trying to detect a pathogen. Now, what does that actually mean? And how does it help you? It means that when you're running a real-time experiment, whether it's, let's say you're doing a gene expression experiment, you need to run a full set of standard curves, which is always should be done in triplicate. And you need to make sure your pipetting is correct, and you need to make sure that your assay is well optimized. However, with Droplet Digital PCR, all you need to make sure is that your assay for your unknowns is optimized correctly, and it will give you a copy number per microliter for that unknown sample, just by doing the partitioning and counting method. We're not going to go too much into detail about multiplexing because that can get really advanced, but of course you can multiplex on our systems to a really high degree. In fact, to the highest degree on the market, up to 12 targets per single well. Sensitivity, detection of rare targets in complex backgrounds. Because of that partitioning, which I'm going to show you in the next slide and a little bit later, it really can make a massive difference to that detection method. And then tolerance of PCR inhibitors. Well, there's 2 points that make it more tolerant to PCR inhibitors than traditional real-time PCR. The first is you're not looking at the exponential phase as you are in real-time PCR. You're looking at the endpoint phase. So if there's an inhibitor present, it just has a slightly reduced effect on your sample. But also the second is because of that partitioning effect, what happens is, let's say you've got an inhibitor present in your sample, heparin, because you're working with blood. What happens is if you have that inhibitor present in your sample, it interacts with the enzyme, your supermix, and it totally causes your real-time PCR experiment to fail because it's free floating and it can destroy all your reaction. However, in Droplet Digital PCR, the partitioning or the droplets are formed before your PCR run. So in some droplets, yes, there's an inhibitor present and it causes those 1, 2, 5, 6, 7, 8, 100 droplets to fail, but it doesn't cause your whole reaction to fail. because those inhibitors have been removed sort of from tited out, I would say. So because of this, it means you've got a way less impact of inhibitors as well. Now, this is a slide maybe you've seen before when it comes to talking about digital PCR. It's called the needle in the haystack approach. So practically, how do I put it before we go into the details about wild types and mutant molecules and all that? If I had to put a haystack in front of you right now, so between me and your computer I assume you're looking at, and there was 20,000 little pieces of hay, and I threw a needle in there, and I gave you a method of finding that needle. I gave you a metal detector, and you came up to the haystack and you put your metal detector and it would go beep immediately because it's detecting there's definitely a needle there. But if I asked you to get the needle for me, you probably would be like, that's not possible, Luke. I'm sorry, that's not going to help. But if I took that same haystack of 20,000 and I divided it into 20,000 and I put a needle in one of those tiny little bundles and I gave you that same detector and put it along the floor, yes, it would take you some time. You'd walk up and down your the floor that you're sitting at, but you would definitely find that needle at some stage and when the metal detector went off, you'd know there's a small area that it could be in and you could quickly find it. Now, that is Droplet Digital PCR when compared to PCR or qPCR. So if we look at the example on the screen, if you've got 40,000 wild type molecules, and this is not something that's unheard of, this is totally normal, probably day-to-day kind of experiment when you're working with mutant molecules, and you've got 40 mutants. Your abundance of those mutants is 0.1%. So really, your chance of your primers and probes detecting that is very, very little, almost none, some would say, especially if it's like a single nucleotide polymorphism and the only difference is your probe has got one different base pair. The chance of you detecting is very little. Now, if you ran that same experiment again in Droplet Digital PCR, you would have 40 droplets that contained those those mutant molecules, which would mean you would have a 33% chance of detecting them. 33% chance, because remember, each of those droplets is checked one at a time, at least in ddPCR, and it's analyzed one at a time. So your likelihood of detecting is 33%. So you can see that partitioning really has drastically increased the sensitivity and the precision of the technology, and that's where its biggest benefit is at. So now we're going to look at just some of the systems that Bio-Rad has offered in the past and sort of positioning them so you guys understand where they all sit. And then we're going to get into the application, which is the really exciting part. So the QX100, some may still have this. This was one of the first systems. Well, this was the first system we ever launched in 2011. So like I said, just over 10 years ago. And as you can see on the left there, at the bottom left, you'll see the number of publications going up. Now, this is something I always love about ddPCR, is it's really, really easy to publish. Well, relatively easy, a lot easier than publishing with real-time PCR, just because of all the statistics required and other aspects. But also, because the technology is labeled specifically ddPCR, it means that anyone running ddPCR is running it on a Bio-Rad system. What does that mean? It means that if you have some sort of study you're looking to run or something you're interested in, whatever it be, maybe you're looking at tropical infectious diseases, you can type in ddPCR, tropical infectious diseases, either on Google or something more specified, and you're going to get publications where people who are running the same system as you, that they generally publish things like their primers and their probes, and you can start to understand how the technology can benefit you. If I type in qPCR, infectious diseases, I've got guys running different systems, barely related to what I know, totally different. Okay, so always really beneficial having someone using the exact same system and technology as you. And you'll see how the publication starts to jump. So moving from the QX200, in 2013, which is a two-color system. A two-color system means it can do FAM and HEX or VIC. And it can also do evergreen instead if you don't feel like using probes. And interesting to know, we're not going to discuss multiplexing, but a two-color system with Bio-Rad can multiplex 4 to 5 targets. and it does this by means of a trademark a patent that's called amplitude multiplexing. It's basically just putting in half and 1 times concentration of your fluorophore of interest. So you can do 4 targets. And then we moved on to the QX200 with the auto DG, referring to the droplet generator, that little system on the left, well, the big system on the left in that case, and in the previous system, that's the manual droplet generator on the right. The manual droplet generator requires you to manually pipette with a multi-channel pipette your samples to the cartridge and off the cartridge onto the plate at 8 at a time. In the automated droplet generator, you can increase your productivity and a lot less hands-on. You just put your plate in the system. It's like a liquid handler, and you select how many samples you want to be partitioned, and it does all the work for you, and it just puts them on a new plate for you. We were the first CE-IVD compliant digital PCR system to market in 2016. Then we had the automated digital system, which was IVD approved. Then we got the auto DG, which was like again CE-IVD marked, and this was for our BCR ABL kit, which was for chronic myeloid leukemia. So a kit that is IVD approved for chronic myeloid leukemia monitoring. Then in 2020, we came to market with the QX1 system, and we can see how the publications have drastically increased over the years. And then just last year, 2023, we launched this QX600 system. The QX600 system, as you likely know or may know, is a 6-color system. That means it does 6 colors. But if you use amplitude multiplexing, how many can you do? If you can do 2, 4 on the 200 system, you can do up to 12 on the 600 system. That means you can do 12 targets per well, much higher than anyone else on the market, and also much higher than what you can do in a current qPCR experiment as well. And then if we're looking at the publications, we can see currently it stands just over 8,300 publications for the technology in 10 years. So really strong growth in the use of it. And it just shows that customers that use this technology, they really like to use it. They feel it's very comfortable and very easy to work with, and really easy to understand and publish and get good results as well. So if we look at the applications field. So what I always say to customers is anything you can run on qPCR, you can run in ddPCR, but you'll get slightly different results, obviously. But not everything you can run in digital PCR can you run it in qPCR, especially when it comes to droplet digital PCR. Why can I say especially when it comes to droplet digital PCR? Because of the partitioning method, that dropletizing. What does that dropletizing mean? It also means you can run things like single cell analysis, because you can dropletize a cell into a single droplet, and other sort of things. There's ways to do your NGS library prep in a ddPCR system as well, because the system is separate. Why would you do your NGS library prep in a ddPCR system over using a qPCR or endpoint PCR system? Well, generally when you run on a qPCR or an endpoint PCR system or endpoint PCR system for library prep, what's going to happen? Enzymes are like everything, they're a bit lazy, and they're going to skew your data to your short reads or your short repeats, and your longer repeats are not going to get accurately copied, right? So your data is going to be all skewed to your short repeats. However, with Droplet Digital PCR, you're partitioning all those your droplets, your sample into your droplet, and then whatever the enzyme is stuck with in a droplet, it kind of has to run the full length. So you find your data is a lot less skewed. So what happens is in Droplet Digital PCR, it's the same workflow as you can do for normal library prep. The difference is you're just going to not do the reading portion. You're going to run the thermal cycling profile because the droplets have been generated, and then you're just going to last the droplets open, and you'll be able to read your sample with your NGS system. So these are just some of the applications. I'm going to start at the top left. Just look at the time. Don't want to take too much. So in the top left here, we've got mutation detection or liquid biopsy ctDNA. And I've got a little example that I'm going to show you in a couple of slides later. But basically what this means is when someone has a tumor, let's say a cancerous tumor. What generally has to happen is the doctor has to do a biopsy, which means they have to put the patient under, they have to do surgery and remove a little portion, and then they can do various tests. They send it to a pathology lab and they do a lot of testing on the sample, which is obviously really important and has to happen. While they're doing these tests, they're trying to see what mutations maybe the patient has, and that will guide them with sort of the treatments that they need to give the patients as well. Now, the limitations of that is that obviously your patient has to be in hospital, it's expensive, it's dangerous, especially as the tumor is closer to more difficult to operate on and those sort of things. It also means the monitoring time is very reduced because you can't monitor your patient consistently. But what happens with tumors is they break off little bits of their DNA. And that little bits of your DNA is those circulating tumor DNA, and that flows in or breaks free and floats in the blood of the patient. And you can use a blood draw and monitor the levels of those circulating tumor DNAs over time. As they increase, you'll notice that maybe there's a massive increase in something like HER2, which is to do with breast cancer, and you'll know it's time to change your patient's treatment because they've developed a mutation and they are no longer able to be treated by a traditional estrogen receptor, which is the normal treatment for breast cancer. So being able to monitor that in a much more quick and accurate method in real time almost, because what's happening to the patients is happening in their blood, instead of coming back every 2 or 3 or 4 months to sort of monitor, means much better detection. And with ddPCR, this is only able because of just that high level of sensitivity, that limited detection being much higher. Mutation detection in tumor samples, yes, we discussed, but we'll briefly touch on that. Methylation detection, really interesting one in terms of methylation. And with methylation these days, there's sort of been a change in how you can monitor that with methylation restriction enzymes instead of using a bisulfate conversion, which is something you can do on ddPCR. So those restriction enzymes are really powerful way to monitor methylation and actually a lot easier than using bisulfate conversion. You can do genome edit detection. So if you're using any sort of editing technologies like CRISPR-Cas, it's really important to know how much of the cells you've transfected have the edit. And with ddPCR, you can accurately do this. How can you accurately do it? Well, one, it's a counting method. So it's going to count your edited genes versus your unedited. But also there's very clever ways of doing your sort of primer probe binding. It's like a drop-off assay. So really important as well. Then we've got microsatellite instability. So yes, that's very important for colorectal cancer, but also just generally for humans as well. Your microsatellites are little bits of human DNA that have repeats. They're, I think 5 or 6 nucleotides long, and they occur throughout the human genome. And what happens is if someone has bad mismatch repair, meaning when their DNA is replicated, and mistakes happen, it isn't accurately repaired. They are known to be as MSIH status, meaning they have microsatellite instability high status. It means that the microsatellites are not stably being expressed, they're changing all the time, they're unstable, which is a great indicator of, chance of developing tumors, other things like that, but also helps in patients' treatment because they have a lot of antigens which are present on their sample. So another really interesting technology, and we have a kit for that called the MSI kit. DNA fusions, kind of straightforward, but really interesting as well. Gene expression analysis. Why would you use a gene expression analysis on ddPCR over using it on qPCR? Well, you don't have to use a standard curve, which could obviously save you quite a lot of money because standard curves are quite expensive. But also, if you're working with very little gene quantity, it's quite important, especially things like microRNA, which is quite limited in quantity as well. Or if you just want to have absolute quantities rather than relative quantities. So for a normal gene expression analysis experiment, you're going to say there's an upregulation of 2 by looking at the difference in the CQ values. Whereas with ddPCR, you're going to be able to say this is 2 copies per microliter, and this gene is 5 copies per microliter. It's being upregulated 2 times. Just more simply put, because you're actually working with absolute quantities. And then copy number variation, I'm going to briefly touch on after this. Viral load and viral load titer, really helpful to know the actual and absolute viral load of samples, determining, and also great if you're ever wanting to make your own standards in your labs. You can actually have a quantifiable amount of, your standard, and you can even, look to sell it or something like that. Loss of heterozygosity, quite straightforward. Residual DNA detection biologics, really interesting. If you're in any sort of biopharma industry, you obviously have to know how much bacterial DNA is in your sample before you sell it for humans, and you have to detect or quantify this, and you can do that with ddPCR. And then wastewater detection. Wastewater monitoring became really big in COVID and still quite popular these days, especially in the EU. And why is it important? Because you can monitor whole areas, wastewater, whole suburbs, and be able to know where your population is in terms of developing diseases well before they come and present to the hospital. So really interesting as well. Now, if we look at just 2 brief examples, because I don't have much time and this is just an introduction, but we're going to look at copy number variation in real-time PCR versus ddPCR. So on the left is the ddPCR results. You can see there's, I think, 1, 2, 3, 4, I think there's about 8 samples. And on the right, you can see, okay, sorry, I should have just read 8 wells, so that obviously implies 8 samples. And then on the real-time PCR, also 8 wells. And you can see here how stable at yes at 1 in qPCR, 1 copy number. So remember copy number variation, for those of you that don't know, copy number variation is really important when it comes to how many genes, how many copies of certain genes an organism has. Because as the number of copies of those genes increase, so does the expression change, okay? And that obviously has a very material impact on that organism, how it does anything in life really, but how maybe it handles certain drugs or certain foods or certain treatments all have a big effect on the number of copies that that gene has. So it's really important to accurately be able to quantify that. And the way you quantify copy number variation is you take your gene of interest that you want to know the copy number, and you take it over a reference gene, which you know has a copy number of 1, means that reference gene is only has 1 copy on a chromosome, and you compare the 2. Now, if you look here again, sorry, going back to this example, you can see how as you get into a much higher copy number state, for example, 4, 5, and 6, how poor qPCR is at accurately determining that copy number. Between 4 and 5, it doesn't even know the difference, and you can see how high the error bars are. Whereas when you compare that to ddPCR on the exact same sample, you can see how accurately those are measured in between. And why is that the case? Well, it becomes technically quite difficult to call a difference of 5 to 6. Why does it become technically difficult in qPCR? Because in qPCR, all you have to measure is a CQ value or a CT value. And what is a CQ or CT? It's a cycle number. But let's say if you have a cycle number of 25 and then another cycle number of 26, if I go from 25 to 26, what does it actually mean happened to my genes? It means that they've doubled. So it went from 10 to 20 or whatever. So anywhere in between, for example, 5 to 6, there's no doubling. There's no 5 to 10. So anywhere measuring in between becomes very technically difficult. So if you're looking at a qPCR experiment between 5 and 6, your CQ value should only vary by 0.25. So you're really going to actually have very little idea of changes in copy number. So we're just showing you the benefits of using ddPCR for copy number variation as well. And then practically speaking, what does copy number actually change? So if we look here at this article, maximizing antibody production, voila, So some sort of biopharma production requires gene editing. The copy number of the heavy and the light chain sequence can affect the productivity, so just how much they're making. By using ddPCR, they can determine the best copy number for the light and the heavy chain antibody inserts. Okay, so what they've done is, remembering with qPCR, they would never really be able to determine the difference between these copy number states, but with digital PCR, they can absolutely do it. So they did this. If you see on the left, they did a cell line and they transfected the cell line with plasmids that contain the light and the high and the heavy chain inserts. And the ddPCR showed the results of a mock transfection, the native copy number, and one which is after transfection. And then on the right, you can see they compared these antibody productions of the various combination of inserts. And top is the Western blot showing the heavy and the light chain protein abundance. So you can just see how this copy number variation has actually played a huge role in the quantity of antibodies that they're actually able to produce in this industry. So a real material impact. But this is just one example in biopharma. Of course, copy number variation plays a massive role in a lot of human and various organisms' life cycles and studies as well. Now, if we look at something totally different, which I said I'd touch on in terms of personalized cancer care, if we're talking about cancer and drug resistance, it's always hard to accurately put, because it's always large changes. So the use of a tumor genotype is used to guide patient treatments and is a routine in a variety of cancers like melanoma, colorectal, and breast cancer. And if we look, for example, at something like melanomas, they have around 50% of them have an activating mutation in this BRAF and V600E. So that's on the right side here. Now, these BRAF and V600E, they account for around 80% of the melanomas that get these mutations. So we need to do, if we're doing personalized cancer care treatments, we need to detect what? We need to detect the BRAF mutations, or the V600E mutations, right? Now, if we look at the samples that are used in for cancer samples, you can do large-scale discovery tools that are using like an NGS or an array. They're really successful to profile the cancer genome, and they're great for validating the samples. But the most important thing is that the samples that we generally use in cancer are very difficult to work with. They have really huge technical limitations. If we think of FFPE, which is a lot of sample that we have left, it's damaged. There's small fractions are amplifiable. If we look at cfDNA or cell-free DNA, it's fragmented. There is a large number of non-viable or non-amplifiable tissue, so really difficult to work with samples. And then if we talk about this liquid biopsy, which we briefly touched on as well, just that fact that, you know, it's a largely non-invasive procedure and it's more representative of the overall body rather than a biopsy of a tumor in a single location. And also, you know, multiple biopsies over time are impractical, expensive, and are dangerous for patients. So if we look at the examples here, we can see that cell-free DNA, which is 99% wild type and 1% tumor DNA, means that the chance of you detecting that less than 1% tumor DNA in a normal PCR or NGS or something similar is really, really little to none. So what does that actually mean? If we look at the example here for PCR, so this is not ddPCR, this is just for PCR. Normally, when you're trying to detect a mutation, you have the same primer set, but you have a different probe set. Well, almost the same probe, maybe one nucleotide difference. Now, what's going to happen is your primers are all going to bind and your probes are going to bind, but because your detection of your tumor is so limited, 1 in 100, you're never actually going to get any results because your reactions are generally depleted before you're even able to read. any calls. So it just shows you again how much higher sensitivity in using ddPCR is for this. So if we look at mutation detection and low frequency, if we're looking at FFPE, cell-free DNA, and circulating tumor DNA samples, in Sanger sequencing, it's around 20%, and NGS maybe around 5% sensitivity at low frequency. qPCR is around 2%, and ddPCR you can get down to less than 0.01%, and these have been practically done. And again, to quantify and analyze rare cells is really a huge tool for ddPCR as well. So to wrap up basically what we've discussed, just to highlight that these are basically all the super mixes and things that are required in a ddPCR in a workflow. It's basically just choosing your super mix, the same as you do with qPCR. You've got your plates and your plastics, same as you do for qPCR. The only difference is these oils and these cartridges, and these are responsible for the partitioning effect. And then on our website, we have an assay design tool for anything that maybe you want to design, a copy number variation assay or something similar, or just running a qPCR assay or similar, that are available as well. And then these are our different assays that we have available in terms of the current ones. You can see there the kits are ones that include supermix, and you can see there's for multiplex mutation, which is cancer, residual DNA quantification for biopharma, copy number determination kits, library quant for Illumina, mycoplasma detection, HEC quantification and sizing, really important for biopharma, gene expression, cell and gene therapy, genome edit detection and mutation detection, all really important. Now, just to finish off, obviously this webinar has been supported by Lasec and with the help of the staff at Lasec. And the biggest thing for many of you that are in attendance today is, yes, wonderful, Luke, thank you very much for explaining ddPCR to us. Well, maybe you don't know anything after I've explained it to you and you're still confused. But how do I actually get the technology? Who's going to actually support me at the end of the day? All those kind of questions. So Lasec is our partner, distribution partner and partner in South Africa, and they support a large portion of the African continent. Obviously, Lasec has their own application specialists that are specifically working with Bio-Rad portfolio only. They have Bio-Rad certified and trained service engineers. They have a full team of sales representatives that support you guys out in the field. And then on tier 2, of course, if there's issues or there's a more technical question or you're not getting the answers you need in terms of an application you're running, I'm always on call. I'm happy to come out. I visit customers weekly, and I have with me a team of regional support specialists based in Europe who always answer my questions. We have a center that's specifically, as you can see, most of the slides that I showed you would say something about digital biology center. We have a center that's specifically focusing on ddPCR, new and novel applications. I have dedicated product managers and support team in the technical support staff as well. So I want to say thank you so much to all of you that attended this brief webinar, and I hope that some of the answers you were looking for, you were able to find. And for those that you weren't able to find or you didn't quite get, please feel free to reach out to myself or to Michelle, or the team at Lasec, and we'll absolutely do our best to assist you. So thank you so much for your time today. I really, really appreciate it.